Welcome back, everyone, to the Academy of Integrative Health and Medicine's weekly wellness webinar series. We are just thrilled to be back. We just came off of our annual People Planet Purpose conference. It was truly amazing. And we are so blessed to be here. We've had so many people waiting for this third talk in a series of 3 from Dan Handley. Dr. Handley is here with us today from Southern California University of Health Sciences, speaking about astounding successes using gene therapies. So as people are coming into the session, just for those of you who are new to the Academy of Integrative Health and Medicine is a global interprofessional integrative health association working to transform healthcare, body, mind, spirit, community, and planet. And if this is your first time here, you can go to our YouTube channel at youtube.com/AIHM Global. If you subscribe to our channel, all of these weekly sessions are always there recorded for free, and you can access them anytime. I'd also like to invite any of you, if you are for the first time thinking about joining our Integrative Health and Medicine Fellowship, you are able to get a discount off of tuition if you have not applied yet, and you can use the code, let them know that you attended a wellness webinar and that's where you heard about it. Our 2-year program, the next cohort will be starting in April of 2022. So please reach out to us and learn more about the program. It's a phenomenal interprofessional program that results in national board certification eligibility for MDs and DOs. So we invite you to come and learn more about that. So today I'm so thrilled to welcome back Dr. Dan Handley. Dan attended Johns Hopkins University, Carnegie Mellon University, and earned his PhD in human genetics from the University of Pittsburgh. He is the director of the Clinical and Translational Genome Research Institute and the director of the Master of Science in Human Genetics and Genomics degree program at the Southern California University of Health Sciences, one of our annual supporters. He also serves on the board of directors of the Predictive Oncology Incorporated. And Dan, we're just thrilled to have you back for this third part of your 3-part series on genetics. Just very excited about this talk. So thank you for being here and welcome.
That's my pleasure. Thanks for hosting me and having me back. I hope everyone is excited about this topic. So with no further ado, I'll get started. Okay, just gotta bear with me for a second because I have to do this sharing part. And beginning. And then I have to swap. Okay, so do you see the whole screen now?
It's a little bit blocked on the left side again. I don't know why it keeps doing that for you. Are you able to zoom out? Oops.
Let me share it again. Is that better?
1 second, it's showing up. Yes, now I can see. Thank you.
So is it?
Yeah, that looks great. Great.
Great. So I'll be talking today about astounding successes in using gene therapies. Something that we're going to be hearing a lot more about in the future, I presume. Already been introduced, but let's see. Just a little bit about me. We've already talked about most of this. Was also, in addition to other things, I was a researcher at the UCLA School of Medicine. I was a founding technology innovator, developing methods for doing PCR testing at the National Genetics Institute, which is now a subsidiary of LabCorp. And I was also senior researcher at Procter & Gamble. and I did my postdoc in gay women's research institute in maternal and fetal genomics health. I'm also the editor on editorial board of 2 journals, Life Research and Clinical and Translational Sciences. And also, I have to disclose that I'm a member of the board of directors of Predictive Oncology, which is a publicly traded company on NASDAQ. The learning outcomes for this presentation is want to cite prominent examples of diseases in which ex vivo gene therapies have been successful in early clinical applications. And I'll discuss all of that, obviously. Explain the major steps involved in ex vivo gene therapy, discuss or distinguish between ex vivo and in vivo gene therapies, list the major categories of in vivo gene therapy delivery systems, list the risks associated with therapeutic somatic cell gene therapies, and discuss the geographical restrictions on germ cell gene therapies, which I think you'll find very interesting. So don't worry if some of these words are mystical to you right now. I'll explain everything here. So gene therapy isn't science fiction anymore. So it's always been part of science fiction movies, and there's a lot of hype that's been around it for a long period of time. But now it's actually coming into fruition, and it's on the verge of being used in certain clinical applications. So I'll explain that. But certainly for some inherited diseases and acquired diseases such as certain cancers. And so I think you'll be kind of astounded at what's been happening in the field. So first, there are just many different methods for gene therapy, and they all have challenges. So nothing's 100% perfect yet, but they're making great strides in successes in using these gene therapies. So I'll give you an overview of the gene editing machinery, and we'll focus on one of them in particular. But just to be complete, these have been around, some of these, for a decade or more. There's meganucleases, there's ZFNs, which is zinc finger nuclease. And the idea is that if you know about how genes are transcribed and you have promoter regions of genes, the promoter that attaches, finds the right gene to attach to, has something called a zinc finger. And that's why actually zinc is important in your diet. It's one of the reasons. But it's a conformation of the molecule that allows it to find a specific place in the DNA. Well, in this context, that principle is used to find a specific place in DNA, and it's attached to a nuclease. And a nuclease is an enzyme that cuts DNA. There's also talons, which everyone in science loves these acronyms, right? So these are transcription-like effect nucleases. So they do something similar to the zinc fingers, but these are proteins, and they're designed in the laboratory to look for specific places in a genome to cut. And the first three, so A, B, and C, have had a lot of difficulties. They're still not completely discarded, but the real exciting one is the CRISPR-Cas9 family of gene editing tools. And I'll explain it really briefly. I won't go into a lot of detail, but what CRISPR stands for is clustered regularly interspaced short palindromic repeats. And palindromic repeats is, palindrome is like madam, I'm Adam, right? All these palindromes that they read the same backwards as they do forwards. So they're nucleotide sequences that read backwards and forwards the same. And then they also are attached to something called a CRISPR-associated nuclease. Now, obviously, nuclease I just described it's something that it's an enzyme that will cut DNA at a certain place. And so I won't go into the details of how double-stranded DNA is repaired, but there are over 200 enzymes or different types of proteins that the human body makes, and 200 genes that that code for those proteins. And so, like over 200 of them. BRCA1 and BRCA2 are prominent examples of enzymes that help repair DNA. And when DNA is replicated, it looks for errors and in the duplicating of the genome and repairs those errors. And so that's why if you have a deletion or some sort of mutation, loss of function mutation in BRCA1 or BRCA2 or both, that now you have the higher chance of genomics instability. That's why it increases the risk of cancer, because you don't want mutations introduced into your cells. So now we have repair mechanisms also for double strand DNA breaks, which are the worst. So if you have a single strand and it breaks, you have the other strand that can keep it together, and then an enzyme can come in and repair that single strand break. Now, when you have a double strand break, and those are, well, they're induced by a lot of different mechanisms, by oxidative damage and also ionizing radiation. So X-rays, CT scans, gamma rays, any sort of ionizing radiation. One of the things that they tend to do is they're so powerful that they actually break apart the whole DNA strand. So you have double strand breaks. And then there's two different mechanisms for repairing those and putting those strands together. Now, the problem is that in some instances in the natural environment, it doesn't know where to put the two ends. And so that's how you get some chromosomal rearrangements and things like that. So I won't go into more detail about that. But this CRISPR-Cas system utilizes the natural repair mechanisms that our bodies have to repair double strand breaks. And so what it does, that's how it works. So you have guide DNA or RNA that's complementary to the sequence that you want. And these could be, many nucleotides long, which means they can be very specific. And so they will look for a specific place in the human genome, and when they match the complementary strand on the DNA, then they bind tightly. Then they have the nuclease, and then they cut it. And then if you want to insert DNA, if you want to insert a gene, you can put your gene in with that machinery, and the natural repair mechanisms will put that gene in or repair that. So you need one on each end, obviously. So that's in a nutshell how that works. But it's been very, very, very successful. And it's so huge that it's been used in a lot of different diagnostic or therapeutic instances. So it's been studied in the laboratory for different treatments or cures. Certainly inherited monogenetic diseases I'll talk about, blood disorders I'll talk about, various cancers, but also it's being investigated for allergies, immunological disorders, Duchenne muscular dystrophy, cardiovascular disorders, neurological disorders, liver, cystic fibrosis, and even viral infections. So there's a lot of excitement around this, and so much excitement. The discoverers of this system, Jennifer Doudna and Emmanuel Champatier, both shared the Nobel Prize for chemistry for the CRISPR-Cas discovery in 2020, which is in the Nobel Prize community, being awarded the Nobel Prize that quickly indicates the importance of this discovery. And the CRISPR-Cas system's been used all over. And I might add, they're actually easy to do, relatively easy to do in the lab. So it's not really sophisticated technology that's required to do this. So one feature common to all gene therapies is that they allow you to edit, replace, or silence a gene, depending on what it is that you want to do. So, or repair, if it's a single nucleotide mutation, you can do that. So let me start this video again. So you can edit So you can do a single nucleotide edit. You can replace a section of the genome, including entire genes, or you can silence a gene. If a gene is producing a gene product that is causing a pathological phenotype, then you can silence it. So there's a lot of applications for this. Also, in this topic, we need to make a distinction between gene therapy on somatic cells and germline cells. That's going to be very important. So somatic cells make up the bulk of the human body, right? And so they're essentially fixed. We don't pass them down to future generations. But germ cells, germline cells, sperm and egg, the DNA in them, if that's altered, that can be passed to a baby and then future generations potentially. So there's a huge distinction between the two. So again, during meiosis, if you alter or meiosis, or actually after the egg is fertilized, and you alter the DNA, that when it becomes a full individual, that whatever you did to the DNA can actually be passed to future generations, which has a lot of practical and ethical and legal consequences. And so it's very controversial, poses many ethical issues, and the web link here is actually a tracker that shows where different countries are on the restrictions on germline editing. And it's not illegal in the United States. Right now, you just can't use federal funding for doing germline editing. But if you have private funding, there's nothing to stop researchers from tampering with an embryo's genetic material. And the first time that it's been reported being done, it was 2018. There's a Chinese researcher that did gene editing on an embryo, and it ended up being live births of twins. And what he tried to do was he knocked out a certain gene called CCR5, and that's a receptor that allows HIV to infect a cell. So apparently he was well-intentioned. He wanted to make the person that would be born, which ended up being twins, but he wanted them to be completely resistant and not being able to be infected by HIV. The Chinese government didn't take too kindly to it, sentenced him to 3 years in jail, started a lot of debate about how it should be regulated. And different countries have different regulatory requirements. And I'll stop this share and then try to go to that just really briefly. But I believe the link is in the chat. So can you see that?
Yes, we can see the website.
Good, good. So this covers all sorts of things: therapeutic, stem cell, germline, embryonic gene editing, gene drives, which is beyond the scope of this talk, and then agriculture as well. So this is an interesting website to browse, and I won't go through the whole thing, but here it shows you where the geographically what kind of restrictions are imposed on gene editing. And here again, it's like a little map, and it shows you where it's mostly prohibited, and other places it's not. So there's no worldwide consensus. And so there are places where researchers, with using private funding, if they wanted to make quote unquote designer babies, if they wanted to edit the genomes of embryos and with the intention of having live births, there are places that they can go in the world where they wouldn't have any trouble doing it. It wouldn't be illegal. And like I said, it's not even illegal technically in the United States. Most researchers, the vast majority, have voluntarily, vowed not to do any sort of embryonic gene therapies. But in terms of the legal restrictions, it's very spotty throughout the world, and it's not very strict. So I'll go back to the presentation here. And again, I urge you to look at that website at some point and browse that. And so now, can you see the
Yes, looks great.
Great. Next slide. Okay, so somatic cell gene therapies are a lot less controversial, obviously, because if you alter the part of the genome of a human being who's already a child or an adult, that it's not going to be, it doesn't affect the germline cells, and it's not going to be passed to future generations. So this is where the therapeutic gene therapies are getting the most traction and are getting a lot of attention. There's some limitations, and we'll get into some more of those a little bit later, but they're best suited right now for single gene disorders, so monogenic disorders such as sickle cell anemia, but they can also be used for certain cancers, and I'll talk about those as well. Genes or disorders involving multiple genes are difficult to tackle at this point. People will be looking into those in the future, but right now it's really single gene disorders, which there are a lot of. The other distinction to make is between ex vivo and in vivo gene therapies. And that's another, it's a huge distinction. And so we'll be talking about ex vivo mostly, but also in vivo. Ex vivo has had the most successes. And what it involves is taking cells out of the body and then doing your genetic, therapeutic genetic editing inside those cells. And usually they're progenitor cells, or they're stem cells for a particular type of tissue. You take them out and then you genetically alter them to correct whatever the defect is, and then you put them back into the body. And so that's had the most success so far. In vivo is a little more difficult, because the idea is that you actually put the CRISPR-Cas in any other things that you need, ingredients in there. You put the genetic sequence or whatever it is you're trying to do. You actually put that in some sort of delivery vehicle called a vector, and you put that into the body. The advantage of that, and why people are still looking for that, is that they're looking to make progress in that area, is because you can target tissues that you can't target with ex vivo gene therapy. So ex vivo is really more suited for blood disorders. But if you're trying to do something that is therapeutic for the neurological system or for the liver or something like that, another organ, then in vivo, in theory, would allow you to design something that would only go to those tissues and do their gene editing. So one of the big successes is in sickle cell anemia. In sickle cell anemia, if you remember your basic biology, it arises from a mutation in a gene called HBB, and that codes for a portion of hemoglobin. Now, because of that mutation, then that gene produces an abnormal protein, and that actually causes a malformation of the red blood cells, which means it's not efficient at transferring oxygen and carbon dioxide. So people can have, there's many different forms, but it's typically very debilitating. Some people, it's very, very severe. And up to now, there hasn't really been many effective treatments for it. So essentially, people have had to suffer with the disease, for life. It's an autosomal recessive trait, which means it's passed. Both parents have to have the trait, and for you to get the sickle cell anemia, so both parents have to have that. And so that's another reason that if you're unlucky enough to get 2 copies of the mutated gene, that it's imperative or very valuable to actually have that corrected. And so what they do is they take the red blood cells from the bone marrow, so the progenitor cells or stem cells that produce red blood cells, take it from the bone marrow, and they use a virus or some sort of vector to insert a normal beta globin gene, so one without the mutation, or they can correct that single nucleotide mutation to produce a normal gene. And then they inject that back into the body, into the bone marrow, and then the bone marrow starts producing normal blood cells. And so they're normally shaped, they function correctly. It doesn't correct all of them, but it's enough that these people effectively are essentially cured, the ones that for which this works. Now, it doesn't work in all patients, and that's one of the problems. But the ones that it does work in, it's very successful, and they've actually gone to phase 2 clinical trials now with this therapy. And it's also been used in the laboratory for a lot of different serious diseases that really don't have a lot of options for therapeutic interventions. Unfortunately, in some of these trials, they've done small clinical trials. You see down near the bottom of the list is Xlinked severe combined immunodeficiency in the gene that's associated. It's a monogenetic disease, obviously. When they've tried to correct that through ex vivo gene therapy, in one trial they had 20 patients. 17 were essentially cured, but they also had several that developed leukemia, and one died. And so kind of take with the good with the bad. And the idea is that this needs to be perfected so that you don't have those those severe effects, and you certainly don't want deaths from this. But the potential for therapy is so great that a lot of work is being put into this to make sure that it's safe. And it's also been used, ex vivo gene therapy, to treat certain blood cancers as well. So one in particular is B-cell acute lymphoblastic leukemia. And so what they did was they've taken some T cells, and in the laboratory, they modify them genetically to produce a cell surface molecule called a chimeric antigen receptor. So this is called CAR T therapy in short. And so in this little diagram, the T cells are removed from the patient's blood. The gene that encodes for that CAR, that chimeric antigen receptor, is inserted. The cell starts producing that. And there's a little, in number 3, there's little crosses there. Those are the cell surface receptors that are specific to the leukemia cells. And then they grow those up in the laboratory, so there's lots of them, and they infuse them back into the patient, and they start attacking the leukemia cells. And that's been very, very successful. So that's another way that gene therapy is being used. Now, and they've actually had patients that their leukemia has gone into remission after receiving this therapy. So there's a lot of hope that this type of gene therapy will bring a lot of relief and improved quality of life for a lot of patients, and potentially cure them for life. Now, one of the problems is not everybody responds well, and researchers don't know exactly why that is. Some people, when you test them in larger populations, anytime that you inject, whether it's ex vivo or in vivo, anytime you put something into the body that has been modified, or if you put any sort of gene editing material into the body, there is always the risk of having a cytokine release syndrome, or otherwise known as a cytokine storm. So now I'll talk about in vivo gene therapies, because they do have some benefits. Like I said, they can reach other organs that you can't do with the ex vivo gene therapy. And so there are a lot of different ways to try to get those materials, CRISPR, Cas, if you're using that, into specific tissues. So there's different viruses. Adeno-associated virus is one of the most widely used and most promising, because it's essentially a variant of one of the viruses that causes a human head cold, and that version really doesn't make many symptoms, and so it's less likely to evoke a strong immune response, which is one of the problems. There's also nanoparticles. There are a lot of different variations of using nanoparticles and other methods to mechanically shoot in little particles that contain that gene editing machinery. So again, with ex vivo and in vivo gene therapy, there's a big difference between the two. Both the most promising seem to be the CRISPR-Cas systems, put them to use. But again, there's a lot of hurdles, and the biggest issue to be resolved is ensuring that any sort of new genetic material gets into the human genome correctly and does not cause harm. And so, again, one of the major ways is that it causes harm, and they've actually had deaths and they've had to stop clinical trials, is that they can induce that cytokine storm. And then in this diagram is actually cytokine storm depiction caused by COVID-19, but it's essentially the same mechanism when you get the cytokine storms, and they're frequently fatal. And so that's one of the things that's kind of holding in vivo gene therapies back, especially. The other thing I mentioned before about some of the patients getting cancer, well, there's two mechanisms of why that would be. One is if any of your gene therapy machinery has off-target effects, which means they don't cut where they're supposed to. And if you're especially if you're putting a gene, inserting a gene that's supposed to be therapeutic, there's two things that can happen generally, two adverse things that can happen. One is that it goes into a proto-oncogene. And if you know of the genomics of cancer, proto-oncogenes don't do anything. They don't spur the growth of cancer until they're activated. Now, when you put a gene in there and you put the promoter region of the gene in there, then that gene is going to be expressed. And being expressed means that a transcription factor and all the machinery for making the messenger RNA attaches to the promoter region, goes through the gene that you inserted, but it can also go through the oncogene. Now the proto-oncogene becomes an oncogene proper, and it starts producing generally growth factors, an abnormal amount of growth factors. And so what does that do? Well, you have a lot of growth factors that are being produced. It's signaling cells to proliferate, to divide, and to grow, and to just keep doing that endlessly. So it's like there's no brake. There's only the gas pedal being pushed down. There's no brake, and so that starts the proliferation of cells, and that's the step that one of the steps that initiates a cancer. The other one is that if you put in a gene into a tumor suppressor gene, the tumor suppressor gene is what it sounds like. And there are genes like actually BRCA1 and BRCA2 are considered suppressor genes as well. But there's a lot of like these genes. TP53 is a big one. It's found mutated in over half the cancers that's been tested. So if you're unlucky enough to have the DNA inserted where it actually stops the tumor suppressor gene from working, then that can initiate cancer as well. So there's two mechanisms there, and that explains why some patients, because of off-target effects, can develop cancer. And so that's where the focus is. A lot of the focus is preventing cytokine storms, understanding it better, but also trying to eliminate off-target effects and making sure that these gene editing methods target only the specific place that you want to target and nowhere else. So, which I pretty much described here. So, improving the accuracy of CRISPR-Cas9, and there's a lot of research in this area, a lot of variations, many variations of this, since it's the first discovery of the machinery. In summary, is that germline gene therapy is problematic for a lot of different reasons, ethical reasons and practical reasons. And so certainly a lot of people object to the idea that you can manipulate the human genome in the germline or in an embryo for various reasons. One is they don't want to start the precedent of people wanting to design their own baby, right? So if they think that the ideal baby or the child they want is very athletic and has blue eyes, there's a lot of ethical issues concerned with that. Well, one is only the wealthy could afford it, but also the baby who was ultimately born would not have informed consent. So even if someone has if they do in vitro fertilization, and when they do that, they typically at the blastocyst stage, they'll take out one of the cells that doesn't harm the developing embryo at all. Take one of the cells out, and they look for certain genetic defects, and then the parents can decide if it's serious enough to abort that fetus or not implant, more commonly not implant that embryo and look for a different embryo that does not have that genetic defect. But that's to prevent disease, not to enhance a person. But in either case, even if someone has a disease or even a trisomy like Down syndrome, which is 3 copies of chromosome 21, a lot of people say that even in that case, that the baby doesn't have the ability to have informed consent to have something manipulated on them in their bodies, even if it's to make them healthier, because they could have full, enriched lives, without having their genomes manipulated. And so, it's a philosophical issue, an ethical issue is, what do we consider to be healthy and normal? And people who are born, if someone manipulates their genomes, again, they don't have informed consent. And so if they're told that they were genetically altered, what can they do about it, right? And so there's big philosophical debate there. And the other part is that if you genetically alter a person and they go on to have children and grandchildren, and they start um, you know um, you know a population of people. I just call it the founder effect when it happens naturally. And so you're getting more and more people over many generations that have that genetically altered material in them. And again, now you have a population of people that didn't have informed consent. That starts growing up throughout the population, and we have no idea how what the ultimate effects would be over many generations, and a lot of genetic admixtures between populations. So a lot of issues. And a practical issue is when you do germline gene therapy, or you're doing therapy, some sort of gene therapy on an embryo, that it's very possible that you're actually altering something, and that the big fear is that you can activate a proto-oncogene or inactivate a tumor suppressor gene, but there's a whole lot of things that can go wrong. And who wants to take a chance? You want 100% probability, ideally, that the child will be born normal, a healthy child, and not because of some genetic alteration, having some sort of just terrible defect or something that causes them to be stillborn, or to be to have serious problems and a low life expectancy. And so that happens naturally, and we researchers are looking for ways to alleviate those kinds of disorders, but you certainly don't want to induce them artificially. And so that's a practical reason that that there's a moratorium, a voluntary moratorium on that type of gene editing. But again, if someone had the money and they had the know-how, they could go to another country and start doing that. Somatic gene therapy, again, has the most potential so far. We've seen a lot of successes, but some challenges. And certainly, you don't want to have any sort of therapies where that there's a real risk of harm, up and including death, right? And so again, we don't want to do any sort of intervention that will result in the death of the patient. So that's why the focus again is on preventing cytokine storms, and also preventing on or off target effects. And so that despite all these challenges, there's a lot of research going into this area, and it's progressing very fast, and people are coming up with more and more effective ways of altering the CRISPR-Cas system, so it's much more accurate, and and you're seeing fewer and fewer off-target effects. So it seems like we're going to be hearing more and more about these types of therapies in the future. So I've already gone over this several times, so I don't need to go over this in the summary as well. And so I have some references and further reading if you're interested. There's certainly plenty of things you can read online, a lot of papers you can look up, peer-reviewed articles. It's also, there's many different areas which I've discussed, and there's just a lot of publications coming out constantly on all these different topics, and a lot of different reports of improvements in these types of therapies. So with that, I'm at 12:45, right on the dot, and I'm happy to take any questions that you may have, but hopefully I can answer. And my share. I see that there are some things in the chat.
Thank you so much, Dan. I had a couple of questions. First, I'm just curious to hear your thoughts about, what this will mean for the integrative health community and how we as practitioners, if there's any specific opportunities or challenges you feel like our community in particular will need to, both be ready for and potentially contribute to in this space in the future.
Well, I think the main thing is awareness, right? Awareness that patients are, maybe seeking these types of treatments, and that there are physicians and researchers that are working in this area. And so, I think awareness and understanding of how these therapies work, their potential benefits, but also their potential drawbacks, would help you be able to counsel patients better. And it's up to the individual practitioner whether how they want to counsel their patients. And if you think that gene therapies aren't appropriate for your patient, um, that's your discretion to to voice your opinion and counsel your patients that way.
I think there will be a tremendous opportunity. I mean, similar to what we're seeing with how social media has kind of moved faster than we have as a society is in regards to laws and protections and, all of those areas. I really loved you kind of highlighting that question, that moral question we'll have around really consent, right? So how can you give consent before you even, for a whole generation of potential children who are genetically modified ultimately? I mean, these are just such huge questions, but I think about how we as an integrative health community and how we might think about what person-centered care will look like with some of these issues around genetics, and how we might begin to just think about how we'll be able to participate in that dialogue. And I think there might be really some advocacy areas, of that we'll be able to contribute to.
Yeah, and one of the things also about the people that participate in these trials, and even eligibility to be in the trials, is that a lot of these people, they've undergone traditional therapies, and this is kind of like the last resort, right? And so, and that's common in clinical trials, right? Especially in oncology, right? And so, people go through, they go through the therapy that their, oncology team recommends for them. They might respond for a while, but then, their cancer comes back, and then the doctors at some point say, "There's nothing else we can do for you, but we have an experimental therapy. Do you want to be involved in this clinical trial?" And so, that's a common progression. And so, a lot of the patients that in the studies that I cited were in that category, that they really weren't responding to any type of traditional therapies, and so this was, kind of a last resort. And the fact that a good number of them actually responded positively is a good sign. It shows it's a whole different approach on the oncology side from traditional oncology, right? So traditional oncology is chemotherapy, cytotoxic chemotherapy, which is awful, right? It non-selectively kills all fast-growing cells in your body, which is why you have these terrible side effects, right? Because your gastrointestinal system and your hair follicles are some of the fastest growing cells in your body, and they are killed along with your healthy, in the cancer cells, right? So cytotoxic chemotherapy, just, I think it's very medieval, but I don't, there are a lot of ways they don't have, replacements for that. And then there's surgery, of course, which is traditional, that goes back centuries. And then radiotherapy, where you're irradiating a person with high intensity radiation, and have gotten very, very precise on and how that's administered. But those are really blunt instruments. And then we have targeted molecular therapies that show a lot of promise, and because they're targeted for specific pathways involved in tumor growth. But there's more work to be done there. But having gene therapies in addition to these other tools, these new tools that are coming out, and immunotherapies, can change the face of oncology and can actually potentially provide people cures. Because right now, if you're using chemotherapy, radiation, or surgery, for the most part, even though there's a fraction of people, and it depends on the cancer too, and in the person, there's a fraction of people that go into remission for long periods of time and could be said to be cured, but a large proportion of the people, a lot of oncologists are saying that we're going to start treating cancer as a chronic disease, and I don't think that's optimal. We want cure to cure people, right? And if gene therapy has that potential to cure someone of leukemia, then that could be the answer. Just solve the problems associated with it. But if it could become a cure without any adverse effects, and certainly without deaths, and that could be a real game changer for world health.
Yeah, we have a great comment coming in from one of our attendees. Angela says genetics counseling may be one area where those with integrative training will be quite useful moving forward, the human care side of those navigating through their treatment options and treatment itself. So, this is a great segue, Dan. I was wondering if maybe you might speak a little bit to the training program that SCU has developed around genetics and, that opportunity for clinicians, because it's really a wonderful program. And I think really will help set up our future integrative community with more specifically trained in this area of genetics clinician.
Yeah, it just so happens I have a slide on that.
Oh, great.
And just one slide, so it can be quick and painless. But yes, we have an accredited Master of Science in Human Genetics and Genomics degree program. It's 100% online, so you can be anywhere and obtain your degree. It's asynchronous. Right now we have scheduled 3 synchronous group seminars, so it's like a little asterisk there, but we may do away with that in the future. But that's not a big burden. Again, you can do it. You can do the program on your own schedule. So if you're a working professional and you can only have some evenings and weekends to work, you don't have to attend any classes. You don't need to rearrange your schedule for anything. You can do it, whenever you want. First thing in the morning, get up in your pajamas and get on your laptop and do your assignments. So it's very, very flexible. It's also unique in that you can earn the degree in 3 terms. It's actually 11 months. Looking at the schedule. So you can get a Master of Science in Human Genetics and Genomics if you go full-time, which is 2 courses at a time. So it's not a huge burden, in 11 months online and at your own, not at your own pace, because you got to keep up with the modules and the courses, but on your own schedule.
Mm-hmm.
If you want to go part-time, which is just 1 course at a time, then you can do that again. I think it's more like 23 months, but you can do it 6 terms. And there's 3 entry points, start in fall, spring, or summer. So we have the next cohort starts on January 11th of 2022. And then there's a summer start as well. So you can start 3 times a year. And then we cover all the latest discoveries and technologies in human genetics and genomics. And so this program was built from the ground up, and a lot of it came from my connections and my experiences in industry and knowing what industry is looking for, in addition to academia, in terms of skills, practical skills, where a lot of programs that are legacy programs, they're teaching the same courses that they did 20 years ago with the same content, right? And so they're producing academics essentially to just follow in the footsteps, right? A lot of graduate degree programs are producing professors, and we know that that's not really where the growth is. And so we emphasize current discoveries, issues like gene therapies, a lot of ethical issues, and practical skills that people can use in their careers, which gives you a lot of flexibility. There's a lot of different opportunities. So it's like instead of being very specialized, like a lot of PhD programs where you're taught to be a specialist in one area, this program allows you to have a broad knowledge of a lot of different areas, which makes you, much more attractive as a candidate, whether you, regardless of where you want to go with your career, or if you want to advance in your current career.
So, I'd just like to say, and I first just thank you so much for this presentation, Dan, and the full series. Any of you who are interested can watch the first two parts, all of them available on our YouTube channel. But I would like to just take a moment and also throw in a few additional words about SCU. The Southern California University of Health Sciences is actually pioneering interprofessional education. And so for our communities that work in integrative health, it is just such an amazing space to collaborate, to learn from different clinicians, whether that's online or in person, and to really be at the forefront of that work that's being done to transform education, as well as, we're all working to transform health and medicine on this planet, but education is so key to that. And Dr. John Scaringe, the president, has really been a leader in that movement and in that work. So it's just such a wonderful organization and institution to consider if you're thinking about getting additional training. And I do just want to thank SCU for being a silver sponsor to AIHM, for being a school-sponsored member. So all of the faculty and students at SCU actually get free membership in the AIHM community. And we just are really working side by side to transform healthcare together. And so, we're looking forward to hearing about the program more, and, in creating a new cohort of educated integrative clinicians, Dan. So I just want to take a few moments also to, and again, I'll be plugging SCU a little bit here, but others as well. At our annual People Planet Purpose conference that just ended, it was online for 3 days. It was absolutely phenomenal. We had unbelievable speakers, really transformational leaders coming together to brainstorm how we can truly transform the work we need to do around health on this planet. And one of the keys to doing that is research. It's research in our area, in integrative health, in systems. And so I just wanted to acknowledge the student winners of the research this year. Jae Hua Park from Bastyr University, Nicole Wells from Bastyr University, and Jamie Kulgis from Southern California University of Health Sciences. And then the researchers this year, the first place went to James Whedon, a phenomenal researcher, clinician, and pioneer, also at Southern California University of Health Sciences, Dugald Seeley from the Canadian College of Naturopathic Medicine, and Michelle Sexton from UCSD here in San Diego. All of them just doing phenomenal work in research. You can see over 40 research presentations that were in competition this year on our website at aihm.org/research. So again, another wonderful reason to think about getting educated at SCU is that their commitment to research and the work that they're doing in that area really helps to move us all forward. So I just wanted to take a few more minutes as we're wrapping up this week. Our next wellness webinar will be our AIHM ACIH Integrative Practice Series. This will be November 12th at 12 noon. And the topic is addressing chronic pain and integrating physical function services into community health centers. And we have a wonderful panel of leaders coming from the Academic Collaborative for Integrative Health that will be presenting. So I really invite you to attend that session, and you can register now for that if you're interested. I also want to encourage you all to save the date for our next year 2022 annual conference, the People Planet Purpose Conference. And this will be both in person in San Diego and then also online. So you will have a chance, we will have a chance as a global community to, finally come together in person once again at the beautiful Paradise Point Resort in San Diego. So put it on your calendar now and remember that we all want to come together next year to really continue to learn together. And finally, I just want to invite you, if you are an individual, if you are an organization, we are growing a global movement. And this last conference that we just ended really began to bring together leaders from all different organizations around the world working in traditional, integrative, and complementary health. And we are really set and poised to bring all of our organizations and clinicians together to strategically transform healthcare on this planet. In doing this, we can synergize and amplify and imagine what we could do, the changes we could make as a unified global community. We have a whole array of organizations coming together to identify key strategic initiatives and move forward collectively key projects that really move all of our work together forward. So I encourage you to join. If you are an organization, consider AIHM organizational membership, reach out to us and get on board as we bring together some of really the global leaders to move forward this movement. So connect with us at the Academy. You can find us on youtube.com/AIHM Global, where all of our weekly wellness webinars are posted afterwards. They are free. You can share them with your community, with your patients, and with other clinicians and like-minded people. You can find us on Facebook, LinkedIn, Instagram, and Twitter at the same handle. Consider joining our community as an individual, as a clinician, as a researcher, as an academic. Join a local chapter. We have an amazing student alliance of student leaders that are interprofessional leaders coming together as well. So however you are inspired, get involved, and we will see you all next week. Thanks again, Dr. Handley, and have a lovely weekend.